everyone, and welcome to today's keynote webinar titled Design and Translation of Ultrasensitive Diagnostics. This webinar is a part of the 6th annual event in the CRISPR virtual event series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. If you have trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits image located on the right of your screen and follow the process to obtain your credits. All right, now let's get started. I'd like to now present today's speaker, Molly Stevens, Kavli Institute for Nanoscience Discovery, Department of Physiology, Anatomy, and Genetics, Department of Engineering Science, John Black Professor, Bionanoscience, University of Oxford. For a complete biography on our speaker, please find it on the left of your screen. Molly, thanks for joining us. You may now begin your presentation.
Hello, I'm Molly Stevens. Really excited to talk to you today about some of our work in designing biomaterials. I'm just going to share my screen. Hopefully you can all see that now. So I'm going to be talking a little bit about some of the work that we're doing in designing and translating ultrasensitive diagnostics. And this is work that's representing a collective effort from my team who come from all over the world. You can see there the many flags from the different countries that they represent. And they also have very many different types of backgrounds. So we have chemists in the group, engineers, surgeons, computer scientists, and physicists, amongst others. We are also working very much in fundamental science, looking all the way through to application, and have founded a number of spin-out companies from the group as well. We work in the area of biomaterials design, and these can be applied in many different ways. So, for example, within advanced therapeutics or disease detection. And we also have been developing new types of characterization tools. And today I'll be focusing mostly on some of the work in disease detection, but also touch a little bit on some of the work in the advanced therapeutics and characterization spaces. So thinking first of all about disease detection. Now, pretty much every disease we can think of, from cancer to cardiovascular disease, to viral infections, antimicrobial resistance, all of these diseases will benefit from having early detection systems in place. So early detection is really, really important. And one of the things that we can do for early detection is to use nanomaterials. And nanomaterials, such as nanoparticles, have actually been around a really long time. So this is the Lycurgus cup from the 4th century AD. You can find it in the British Museum in London. And this cup actually has gold nanoparticles within the wall of the cup. And this affects the way that it interacts with light. If you shine a light inside it, it looks red. And if you shine a light at it, it looks green. So humans have been using nanoparticles for a really long time, but it's much more recently that we've been able to use them in ultrasensitive diagnostics and also been able to characterize them and visualize them with advanced microscopy techniques. So the area that I'm going to be talking about is thinking about how we can use nanoparticles made from different types of materials for ultrasensitive biosensing, including in CRISPR-based diagnostic applications. And the way we think about this in our group is we use nanoparticles we've made from different types of materials. So the core of the nanoparticle might be made from plasmonic materials, for example, or catalytic materials, or even polymer-based materials. And I'll touch mostly today on the catalytic-based material work that we've been doing. And you can then functionalize the surface in interesting ways to elicit the desired biosensing mechanism. And for a lot of the work that we do, we're an experimental lab, but we collaborate very much with the group of Professor Jarovsky, who does a lot of molecular dynamics. And you can use these biosensors for drug screening, but also for early disease detection, which is what I'll focus on today. What you can see here, this is from a science paper we published a few years ago. This is showing you a schematic of the surface of a nanoparticle and how you can decorate that in different ways for different types of biosensing. So, for example, within our group, we've worked a lot looking at detection of different enzymes. So enzymes like proteases, phospholipases, kinases, acetyltransferases. We've also looked at nucleic acid detection and protein detection and small molecule detection. And so the surface of the nanoparticles is very versatile in terms of how you can functionalize them for these different kinds of biosensing applications. The first example I'm going to tell you about is an example, just to set the scene really, of how we can detect proteases inside the body. And this particular application was for cancer, but really it's relevant to any scenario in which you would want to detect enzymes like proteases inside the body. And proteases, as you know, are these enzymes that can cleave through peptide bonds. And the assay that we're going to be reporting here is one that turns the urine blue in the presence of some small catalytic gold nanoparticles that we've designed. So the biosensor looks like what you can see in the top left there. So in panel A, you have a protein core. In this case, this is neutravidin. And this can bind 4 different biotin groups. And those biotin groups are attached to short peptides that have a terminal gold nanocluster on the end. And you can see in that picture that's just popped up some electron microscopy of those gold nanoclusters. Now, when that sensor is all assembled, it's about 11 nanometers in diameter. So that is too big to get filtrated out by the kidney. So if you administer this, for example, to this mouse that has a particular type of cancer, then those sensors are too big to come out through the kidney until the peptides get cleaved by the protease that we're interested in. And that protease can then release those terminal gold nanoclusters that are now just 2 nanometers in diameter, and those are small enough to get filtrated out by the kidney, and they will end up in the urine. And you can then add some chemicals to that, and those tiny gold nanoclusters will turn the urine blue. The in vivo work for this was done in the lab of Professor Sangeeta Bhatia at MIT, and they have a colorectal cancer model. And what you can see here is the urine from healthy mice looking yellow, and then the mice that have an upregulation of this matrix metalloprotease related to the colorectal cancer, it's actually MMP9, the urine is turning this bright greeny blue color after about 1 hour after administration. So this is quite early results, but is a sort of example of how you can design systems that might get activated within the body. I want to talk a little bit about how we can also make diagnostic technologies that are at price points that could really help people all around the world. And this is really important when we think about the burden, for example, of infectious disease that, really, really affects low and middle income countries. So if we can use engineering to help make technologies that are more accessible, easier to use at the point of need, at better price points, this is going to be really, really helpful in democratizing access to healthcare. One of the ways that we think we can do that is by using diagnostics that we can develop so that they can be read by mobile phones. This is an area called mHealth. If you're interested in this, I refer you to our quite extensive Nature paper on this topic. And this is the fact that really mobile phones can be much more useful than just web searches and social media. We can actually also use them in the context of mobile diagnostics and making technologies much more accessible to people at the point of need. And this is really important because there's now over 7 billion global mobile phone subscriptions around the world. So this is a way that we can really empower people and democratize that access to healthcare. Now, what we've shown in this paper here is the growing smartphone adoption across the world, and you can see it's across the world, but it's also in sub-Saharan Africa. And it's not just the number of phones that's growing, it's the capability of those phones. So the fact that those phones can now take better and better pictures, and that's something that we can use to help us make more accessible diagnostics. So, for example, here what we've looked at is some data from Uganda, where we're looking at patients that can either get easy access to a healthcare facility or could get easy access to mobile phone coverage and then have diagnostics read by mobile phone. And not surprisingly, you can obviously access many, many more people if you can get diagnostics developed that could be read by mobile phone rather than people having to go into healthcare. More centralized health facilities. We are not alone in this endeavor. We've set up a center called iSense. It's just been running for about 10 years now. And this is focused on infectious disease detection. We have a very important part of the center looking at symptoms within populations. This is involves people like Ingemar Cox, who are world leaders in this area. They work a lot with Google and Twitter, for example, and have been very active in the COVID pandemic. My focus within the center is around ultrasensitive diagnostics that can be read by mobile phone. And then there's also a real push within the center to look at integration into online care pathways. Now, we have developed a number of different tests, and I just want to show you one that at the time we published this a few years ago was a very, very sensitive test for early HIV detection at the point of need. And it was really a test that was transformative because it could detect the virus itself at very low concentrations, so at very early stages of infection. And the way we did that is to detect a protein on the viral capsid called P24. And we use these very special particles. Instead of biological enzymes like horseradish peroxidase, we use these synthetic particles that can act a bit like horseradish peroxidase and produce these very strong colors by oxidizing a substrate in the presence of various chemicals. And these have a gold core and a porous platinum shell. And you can use them in a lateral flow type test, a bit like the COVID test that we're all familiar with, but they give a much stronger signal. You can functionalize them with antibodies, in this case to the P24 or another antigen that you might be interested in. And we then can, we also have another special molecule in here. So we have a nanobody, which is also going to be able to bind the P24 protein. And this is site selectively biotinylated. So when you mix this with a sample from the patient, the antibodies on the nanoparticle can bind the P24, and so can this nanobody. And that nanobody with its biotin group can then bind very quickly to streptavidin at the test line. And you can then introduce the substrate and the chemicals and generate this colored product, which gives you about 100-fold amplification of the signal. So what you can see with this data here, the black data points are the test before it's amplified. And then once it's amplified, you get right into this acute stage of early detection. And this is a test that we're now developing and going into manufacture, which is called QuickZyme and is applied for many different biological targets as well. With slightly different tests, we can also target different diseases. So this is a different test where we're now looking at serological surveillance of Ebola. We took this to Uganda during the Gulu outbreak, and we're able to detect differences between 3 different strains of the Ebola virus and then capture these by mobile phone. So that's a sort of example of how we can use that mHealth approach for serological surveillance, which is becoming really more and more common now after the COVID pandemic. We work a lot with different partners in Africa, both in Uganda, also in Ghana and in South Africa. This is from one of our recent trips to South Africa, and really we're extremely grateful for the support that we get from all of our partners in the field, and also the sort of advice that we get about what would make the tests as useful as possible. We also can use these very special nanoparticles to help us detect non-coding RNAs. And this is actually where we use these nanozyme particles together with a CRISPR-based assay for pre-amplification-free detection of non-coding RNAs. And so what we're looking at here is it's called an inverse sensitivity test, in that if you have a negative sample, then you see a strong signal at the test line, and a positive sample, you see a weaker signal. So it's an inverse sensitivity test. And the way it works, for those of you familiar with CRISPR-based diagnostics, is we use the CRISPR reaction. And if it's a negative sample, we have our little reporter probes that remain intact, and they can bind our nanoparticles to antibodies on the test line, and we can then see this colored test line signal. However, if we have a positive sample, then the enzyme will get activated by the RNA, and then we can cleave these reporter probes, and they can then no longer bind the nanoparticles, so they no longer bind to the test line. So now we don't get a color change at the test line. This is an assay called CRISPRzyme, and this paper that you can see that was recently published in Nature Nanotechnology is a collaboration with a group of Jim Collins. And you can see that this actually works really nicely for a range of different targets. So on the left here, you can see the inverse sensitivity assay for circular RNA, long non-coding RNA, microRNA, amongst others. And we're getting readouts that are working as well on the right here as for RT-qPCR, but without needing to need sophisticated instrumentation. And we also have a nice dynamic range to enable some quantitative sensing. So it's a really, really nice assay. We have taken this through now to some human clinical samples. So at the top, we're looking here at using the assay to distinguish between two different forms of prostate carcinoma, and this worked very well for this application. And then at the bottom, we use this in the emergency for patients that had been admitted to the emergency room to determine whether or not they had had a heart attack. And again, the results from that, which are shown in detail in the Nature Nanotechnology paper, were extremely promising. So really nice to see that this assay is working in quite complex samples, from tissue biopsies through to human clinical samples. And we anticipate taking this forward now towards manufacture as well. I just want to show a video of our center, some of the work done within our center at iSense. And this is where members of the center have gone out to South Africa, to KwaZulu-Natal, and trained field care healthcare workers out in the field who've run many thousands of tests now and been able to map the results into these data dashboards. And this is by using procedural markers and reader systems that were developed within UCL as part of the iSense consortium. Now, I'm going to spend a few minutes just touching a little bit more in the advanced therapeutics space. So thinking about how we can use materials to influence the way that cells behave. So for example, here, how we can pattern cells in interesting ways for cell differentiation. Also how we can study tissue nanostructure to help us design better biomaterials. So in this science paper here, we zoom right into the nanoscale architecture of bone to better understand the relationship between the organic and inorganic components. And that enables us to redefine to a higher degree the levels of hierarchy within a bone, and also then to design better biomaterial systems. And this is a big theme within our group, this really thinking about and trying to appreciate how much complexity we want to encode into biomaterials design. And I won't have time to cover it in this talk, but if this is something that you're interested in, then I would refer you to these perspective articles that I've referenced on the slide that are all about really thinking about this Goldilocks approach to how do you design materials that are going to be as effective as possible, but also translate in the right way. And another way that we do that is by collaborating with people with many different backgrounds. In particular, I'm director of this UK Smart Materials Hub for Regenerative Medicine, which brings together work from across 10 different universities in the UK. We design many different types of biomaterial platforms, so from hydrogels to porous scaffolds, 3D printed systems, also 2-photon nanopattern systems for use in many different regenerative medicine applications, including in combination with some CRISPR-based approaches. We also use different types of imaging that might be interesting to people. So, for example, we do a lot of work with this technique called Raman spectroscopy, where we can use lasers to measure the biochemistry of cells and tissues and get out a biochemical snapshot of what's going on within those cells. And we've used that over the years to look, for example, at how bone differentiates in this Nature Materials paper here. And then zooming into individual cells to see how they respond to different chemical stimuli, for example. This is actually showing you a false color map of chemistry within the cell in response to strontium ions, and you see really big changes in the lipids within the cells when you do that. And then we developed this framework here called quantitative volumetric Raman imaging, which enabled us for the first time to look at cells within 3D hydrogel systems and look at their chemistry in high resolution in 3D. More recently, together with my ex-postdoc Mads Bergold, we've been applying machine learning to some of the Raman imaging to really speed up the process with this technique called Deeper. And also this other technique framework that we've developed in our group called quantitative Ramanomics, that you can apply to cells, but also in this case, as you can see here, to organoids. And what we can do with this kind of approach, if you look on the right, what we're looking at here are liver organoids, where we can see the nuclei in blue, lipid droplets in purple, and then, the nuclei in yellow, lipid droplets in purple. And then what we're seeing in blue is really exciting. This is actually drug metabolites that is spreading throughout the liver organoids. So you can feed drugs to these organoids and actually understand where the metabolites are distributing within the organoid by mapping the chemistry of these organoid systems, which is very, very exciting. And then finally, we have developed this technique called Sparta that I'll spend just a couple of minutes telling you about. And Sparta is relevant for anyone that is working on nanoparticle-based systems. So whether you're using, these are just some recent papers from our group, but whether you're using, for example, polymer-based nanoparticles or exosomes derived from cells or lipid nanoparticles, Sparta is a technique that for the first time can help you understand the chemistry of these nanoparticles on individual nanoparticles in a high throughput manner, which is a goal that we were very excited about, a challenge we were very excited about trying to solve, and we've now achieved by inventing this technique called Sparta. So Sparta actually stands for single particle automated Raman trapping analysis. And what you can see here is that with Sparta, we can trap a nanoparticle of interest and we can measure its chemistry, and we can correlate this to the size of the nanoparticle, and even do things like real-time dynamic monitoring of chemical reactions. And this is all powered by our own coding and software that we've written in-house. And at the time we published this back in 2018 in Nature Com, this was what we would call a breadboard setup. So it was in a dark room, it was used by the physicists in our group, and it was quite a specialist piece of equipment. And what we've done now in the last few years is make it into a standalone equipment that doesn't need to be in a dark room anymore and can be operated by non-experts at the touch of the button, because we really want this to be used in a widespread way by biologists and by pharmaceutical scientists. So this is what the instrument looks like. It's now launched within a company that we founded called Sparta Biodiscovery. It's now commercially available through our early access program. And you can look at many different types of particles. So on the left, you can see you could look at viral vectors, for example, and extracellular vesicles from cells, many different types of synthetic particles as well. And you can understand their composition and their chemistry, but also, for example, how much drug is loaded there, whether there's any contamination, and so on. And I won't have time to cover it in this talk, but if you're interested in this topic, these are some of our recent papers in the bottom right-hand corner that you can see. So I'm just going to give a couple of examples of how we've applied this in ways that might be relevant to the audience today. And the first example is how we can use this technique to study extracellular vesicles. Now we work quite a bit with extracellular vesicles in the context of therapeutic applications, but they're also very interesting for diagnostic purposes. And in this particular example, we've used the technique to look at them for diagnostic purposes. And in particular, we've looked at exosomes from many different types of cancer cells. So, for example, cancer cells that exhibit different drug resistances and so on. And what you can see is that even without labeling the exosomes, you can distinguish nicely between them due to the different chemical signatures that they have. And this is shown in the data on the left there. And you can also do this dimensional reduction array analysis, which is shown on the right, which gives you kind of a heat map of differences within the spectra of these exosomes when you bin different regions of the spectra. And so you can start to really use this technique to understand the chemistry of the exosomes. And it turns out that you can actually distinguish between cancer-derived or non-cancer-derived EVs with about a 95% sensitivity and specificity. And you could apply this, of course, to studying biology of many different types of vesicles as well. You can also apply it, as I mentioned, to many synthetic particles. This is just one example from a recent paper where we applied it to lipid nanoparticles. And in particular, what we did here is study not only different compositions of lipid nanoparticles, but also how these lipid nanoparticles could be degraded, or not degraded, but could have the head groups cleaved off by phospholipase D. And you can follow that process over time. And this is work by Hannah Barriga within our group. And I'm just showing you two examples here, but really you can use this, as I mentioned earlier, for many, many different types of nanoparticle-based systems. Okay, so very final thing that I want to show you, and I'll sort of round up by talking a little bit about CRISPR again, is how we can also look at interesting physical triggers for drug delivery, whether those be ultrasound or mechanical interfaces or light. And I'll talk a little bit about light in the context of how we can use that to look at CRISPR-based spatiotemporal control of gene regulation. So this is some work from a couple of years ago now, and we essentially developed some photo-switchable CRISPR guide RNAs that would have a photocleavable caging group. So these could be essentially activated by the presence of light. We can then use these to essentially activate genes in vitro or knock out genes in vitro within cell culture systems, and then also even within a zebrafish model. So you can see here the structure of the photocaged guide RNA, to which we can then apply some light and remove those caging groups so that the system can then be activatable. And what we're doing here is essentially looking at cells in vitro and we've got some RFP expression going on that you can see that actually the pattern line in the left-hand image there. So this works really well in an in vitro based system. But you can also use it in vivo in this zebrafish model. And what we've done here is to actually target gene editing for a gene that was responsible for pigmentation. And you can see that we have specifically targeted the light onto one of the eyes. And you can see that the gene editing is able to occur in vivo. And you can essentially control the amount of pigmentation that gets expressed in this system. So this still is an area that we're still optimizing, but it's a nice early look at some of the things that we've been doing in this area. So I'd just like to round off by sort of going back to this summary of the scheme of different work we have in our group, from ultrasensitive biosensing to advanced therapeutics and characterization. We are moving the entire group to the University of Oxford to be based in the new Kavli Institute there, which is a super exciting new institute that really houses medics and physicists and chemists and biologists all under one roof. I'll be the deputy director of this new institute, and please do get in touch if you'll be looking for postdoc and other training positions within our group there. We're going to have absolutely fantastic new state-of-the-art labs, and the entire group will be moving from London to Oxford. So it's a really exciting new chapter for us. I'm very grateful, I must say, for the time we've had at Imperial, but this is a great new chapter. And I'd just like to finish off by thanking again the wonderful group and collaborators and also our funders. And thank you very much.
Thank you again, Molly Stevens, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. Questions submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until November 1st, 2025. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. So until next time, goodbye.